Studies, General Abilities and Basic Proficiency in English, and paper 2 for lecturers, that is the subject which they are concerned with. So you will see that botany paper is given as paper 3 for junior lecturers and as paper 2 for degree lecturers. Both the papers are for 100 marks. There is a demonstration that we can concentrate on at a later time.
Okay. Ma'am, what about chapters, ma'am? Any chapters untai andro important chapters?
If you see the syllabus, both for JL and also for DL, there are 10 topics which are given. Now, you have to understand that botany as a syllabus is very vast, that is roughly divided into 10 major topics or units, you can say, and they are similar for both JL exam and also for DL exam. There are some changes in the syllabus that is there for DL exam, as you know that it is of a higher higher standard. So there are some topics which are extra, but whether it is JL or DL exam, you have to be preparing at the PG level. Okay, so in the 10 topics that we are going to see, we will have the first one, that is phycology, mycology, bacteria and viruses. Now let us understand how these can be prepared for the exam. For example, if you take phycology, there are all general topics. If you see the syllabus here, all the general topics are given, like cell structure, reproduction, whether it is vegetative, sexual or asexual, thallus organization, then criteria for classification like pigments, reserve food, flagella, classification. And when you see here in algae, there are no type studies. So instead of focusing on the type studies like previously how they were used to do, now you can go for general characters in all these classes. So if you compare the characters of these classes, you will be having a rough idea of what is important. Like you have to take these things that they have given for criteria of classification, pigments, reserve food, etc. So if you compare all these classes, you will have a thorough knowledge of what is to be going to come for the exam. And apart from this, in phycology, you can also you should be very thorough with algal blooms, that is general topics like how algae are economically important for us, whether they are causing any harm to us, whether they are causing any useful applications, they are giving useful applications to us, like algal blooms, are they toxic algae, then biofertilizers which are made of algae, then algae which can be taken as feed, and how they are important in our industry. So it is a very wide scope which is given to this phycology, but as I told you before, this has to be learnt in a very general manner. Do not go into the type studies, but compare the special characters or salient features. Now when I am saying compare the salient features, you have to understand that new classification systems also should be learnt here. Okay, the old ones that we were following till now, like Fritsch classification, not only that, but also the new classifications like round, these classifications have to be learnt, and comparative study, as I am telling you from even the previously, I am telling you that the comparative study is much more useful for us in clearing these exams than going into typical type study of the genera. Okay, now the next one is mycology. Under the first unit only, you have also mycology, bacteria and viruses along with phycology. This is a vast area, as I told you before, and this is common both for JL and DL exams. So in mycology also, you have to concentrate on the general characters, then the relationship between the fungal organisms, cell ultrastructure. Now when I say cell ultrastructure, you have to understand that there are unicellular organisms here and also multicellular organisms. So you have to learn both the structures, whether it is unicellular like yeast or multicellular like moles. Then what is the composition of the cell wall, then how is the nutrition. Now there are some terms given here. You see that there are new terminology which is being used here. Now there is called as saprobic, biotropic and symbiotic. Now, any organism which is dependent on other organism for its food, whether it is a parasite or a symbiont, that will be called as a biotropic organism. That is a new term. So, you have to focus on the new terms also along with the old terminology. So, please be proficient with the new terms also. Then the reproduction, the kinds of reproduction, then general topics like heterothallism, heterokaryosis, parasexuality. Then these are the things that you have to be putting a lot of importance on, because the questions might come like, what is the organism in which the heterothallism was discovered for the first time, or who is the person who has done a lot of work in heterothallism. So, these kind of questions might come. So, you have to be thorough with the general topics also in mycology. Then in classification, they have given molecular aspects in classification. So, general classification. Apart from this, you should also learn the new types of classification where molecular aspects like the structure of DNA or the structure of amino acids or proteins and their relationships, these things have to, cladistic type of classification should also be taken into account. Now, then as I told you in phycology, in mycology also, instead of going into the type studies, please keep it in mind that a general account of these classes like our divisions like mastigomycotina, zygomycotina, ascomycotina, these divisions have to be learnt in detail as in a general manner, general account of it. And then the economic importance, I have been telling you before also that economic importance of all these classes is very, very important, like which kind of fungal organism is used in creating a type of cheese, like you have 𝗳𝗏𝗵𝗃 cheese or 𝗹𝗶𝗇 cheese. So, which is the organism which is used in that kind of a production of cheese, or which is the fungus that is used in production of antibiotics, a specific type of antibiotic. And then food, what are the different types of mushrooms or mushrooms which are used as food. So, these are the economic importances, the diseases caused by fungal organisms. Then parasites which are there in it, saprophytes which are causing a great, they are very, very helpful to the environment and nature in breaking down the dead and decaying organisms. So, these are all the economically important fungal organisms which have to be kept in your mind and prepared accordingly. Now, bacteria and viruses also equally important for us, because they are both harmful and useful to us. Microbes are used in medicine, in agriculture, in environment. We have different kinds of molecules, the smallest organisms possible, spiroplasmas or phytoplasmas. These are the organisms which live in the phloem of plants. So, you have to spare some time to learn about the ultrastructure, then chemistry of the cell wall, nutritional types, reproduction and plasmids. These are the things that you have to concentrate on in bacteria. And in viruses, the kinds of transmission of viruses, how the plant viruses are going from one plant to the other, or symptomatology, that is what are the symptoms of a particular disease. How you can recognize that particular disease, seeing the plant itself, how can you come to a conclusion whether it is caused by a particular virus or not. These are the symptoms that you have to learn in viruses. Now, this is the second unit which is there in the syllabus, that is the Bryophyta, Pteridophyta and Gymnosperms. Now, these three subjects are learned all through your intermediate and degree also, but you have learnt in the form of type studies here also. But for preparing for these exams, that is JL and DL, you have to concentrate on the classifications, types of classification, and especially when you come to Bryophyta, the economic and ecological importance. Ecological importance, these bryophytes, they have a lot of ecological importance. You have to learn about these things. Then compare and learn, like for example, in the syllabus itself we have marcantiales, jungermannials, anthoceratales, sphagnales, funerales and polytrichales. Instead of going into the type studies, make a note, compare You just draw in a sheet of paper, write these orders names, write the characters like what is a morphology like in Marcantials, Anthoceratales or Funerales. Like that you write the characters, compare it and learn. This is the best way to learn in short period of time. A lot can be learnt in a short period when you compare things. Then when you come to the next one, that is Pteridophyta also. Now in Bryophyta, evolution of sporophyte is important, and Pteridophyta also heterospory and origin of seed habit. These are evolution of stele. These are the ones which are to be concentrating upon. And in gymnosperms, when you look at gymnosperms, apart from classification and general structure, please go through the gymnosperms which are available in India, especially in southern region, or you can also concentrate on in Telangana areas. Like for example, you have Cycas and Podocarpus and Netum, which is very much available in our state. So concentrate on those orders or families and learn about them. How many of them are there? Where are they distributed? What is the kind of habit they show? These are the things that you have to concentrating upon. Now the third unit, that is taxonomy of angiosperms. This also has an equal importance. Let me make it clear once again that there are 10 units and you have 100 marks. So roughly you can say 10 marks might come from each unit. So each unit is important. There is nothing like this unit is more important than the other one. Each of them have equal weightage, and these marks, how you have to grab these marks without any negative marks, we will discuss that a little later. But in taxonomy of angiosperms, you have to understand that the tools which are used for classifying plants like herbaria or floras, and then we have recent trends in taxonomy, that is what are the serological evidences or biochemical evidences or molecular techniques which can be used as evidences in classifying plants. Then what are the demerits and merits of various classifications, like you have artificial classifications, natural classification, and then there are recent phylogenetic classifications where cladistics are there. So you have to understand the merits and demerits of these classifications. Why we are following certain type of classification, what are the advantages or what are the merits of that particular classification. And then you have families here. There are roughly about 7 to 8 families, and these families can be learnt, as I told you before, in a comparative manner. Learn the families in a comparative manner. What is a habit? What is a habitat? How is the leaf, the stem, root? Are there any special features in these root, stem or leaves? Then what is the kind of inflorescence? Whether the petals are fused or not? These characters have to be learnt in a comparative manner. And please do not forget the economic importance of these families. They are very, very important. As you all know, they are important as food and feed for us. So you have to make a note of the economic importance of these families also. Then you have to understand what are the different types of specimens that are used in naming. Nomenclature of plants also is very important. So you have to know about your ICBN, International Code of Botanical Nomenclature. What is a rank, what is a taxa, what is a hierarchy? Like for example, in the previous paper you have got a question based on what is the hierarchy of the taxa. So you have to learn the proper hierarchy. What are the types that are used, like you have lectotype, isotype, syntype. What are the types which are used for naming? All these things have to be learnt. Then in the next unit, that is in plant anatomy and embryology, both RAM and SAM, that is shoot apical meristem and root apical meristems, these developments, that is how the shoot is developing, how the root is developing, what are the different theories which explain the development of these meristems, where are the divisions occurring, which is the area where the divisions are occurring. All these things have to be learnt. And especially when you are going for competitive exams like these exams, you have to know about the differentiation, how these tissues are differentiating into xylem and phloem. You all know that the mass of cells which is coming out is going to differentiate and develop into tissue systems. So you should know how this tissue differentiation is occurring, whether it is a xylem or phloem or secretory ducts, what are the latticifers, what is the latex used for. These things you have to understand. And then leaf, how the leaf is developing, how leaf is differentiating, what are the different types of phyllotaxy, okay, whether it is opposite or alternate or opposite or whorled, is there any special type. Kinds of phyllotaxy also has a lot of importance in these questions. When you come to plant anatomy and embryology, the other part which is more important is the developmental stages of male and female gametophytes. Now, male gametophyte, you can say this is an vast subject in itself, where you have different varieties depending upon the plant species that you take, how different the structure of anthers are, how the microsporogenesis is occurring, what is the role of tapetum, whether it is a amoeboid type or proper type, how the pollen is developing, how many cells are there in a pollen grain at the time of pollination or at the time of being taken to the ovary, stigma, and then what are the causes for male sterility, how is the sperm of the plants like, whether it is dimorphism is there in the sperm or is it all same, and how is the hybrid seed being produced. Now, apart from this, when you are talking about male gametophyte, you should also learn about the pollen pistil interactions, like how the pollen grain is germinating, what is guiding the growth of the pollen tube, then how is the pollen stored, because we are trying to preserve our natural resources. So, how is the pollen stored? Then there is a lot of nowadays when the pollen grains are being liberated, people come with a lot of allergies, complaints that they are having allergies because of pollen grains. So, what is pollen allergy, what causes pollen allergy, and then the tissue culture part of it, that is how the pollen embryos can be developed. Now, like male gametophyte, even the female gametophyte also, the developmental stages is very important. How the megasporogenesis is occurring, how many types of embryo sacs are there, like is it a monosporic or bisporic or tetrasporic embryo sac, then what is the structure of the embryo sac, how many cells are there in the embryo sac, okay, pollination, pollen pistil interaction, fertilization, then what are the floral characteristics, and pollination mechanism, is it a self-pollinating or cross-pollinating plant, what are the mechanisms or contrivances that the plant has to go for cross-pollination, then self-incompatibility, and the very important thing, that is double fertilization, the characteristic feature of angiospermic plants. So, these are the things that you have to focus a lot on. Apart from male and female gametophyte, under embryology you also have seed development and fruit growth, how the endosperm is developing, whether it is nucellular type or cellular type or halobial type, what are the examples, then how the embryo is developing, that is embryogenesis, then different types of embryos, that is either dicot embryo or monocot embryo, how they are developing, what are the different stages of this development, then how do you classify the endosperm, then what is the condition of polyembryony or apomixis, and then fruit maturation. Whether the seed is dormant, how do you overcome the seed dormancy, whether the bud is dormant, how do you overcome, or what are the methods of bud dormancy removal. Along with this, in DL exam, there is one topic which is added apart from what the syllabus is for both JL and DL, this is one particular topic which is extra, I mean which is added to the DL syllabus, and that is senescence and programmed cell death. So, aspirants of DL posts in Gurukulas should concentrate on this particular topic, that is PCD, that is programmed cell death. How many types of cell death are there? Then what is the importance of PCD in life cycle of plants? What are the metabolic changes which are associated with senescence and its regulation? Then what is the influence of hormones and environmental factors on senescence? So, these are all the things which are extra, I mean which are added for the DL syllabus. So, you have to concentrate on this. Then embryology related to taxonomy. How classes or how family plants are classified. There are some examples where the embryological evidences have been taken to solve the mysteries of the classification in plants. So, that has to be taken care of. Now, the next chapter is, fifth chapter is plant resource utilization and conservation. Now, here you have to understand that the plants are considered as a major resource for our survival. No plants, no survival. So, you have to understand that plants are designated as a natural resource. How they are identified? After identifying, how they are utilized, proper identification, proper utilization, and also their conservation, because if you want to have these plants to be going for the next generations also, you have to conserve them, because we all know that we have been using indiscriminately. So, this will not give us a sustainable development. So, you have to see that plant resources are identified, utilized and conserved. This also is a very important chapter or topic in view of this recent developments. So, you should know about how the food crops or forage crops or fodder crops or fiber crops. In a nutshell, you can say that commercial crops, how they are originated, how they were originated, how they have evolved, and how we have cultivated, and what are the uses of these crops. And then one most important thing is ethnobotany. What is ethnobotany? That is a knowledge of the tribal people about the different uses of plants. You know, they are living in close association with nature, so they know a lot about plants than us. So, the scope and objectives of ethnobotany, that also has to be learnt. Then when you talk about plants, it is not only food and forage, you should also know that wood, timber yielding plants and non-wood forest products such as bamboos, rattans, raw materials, paper making, gums, tannins, dyes, resins and fruits. Now, please understand and learn the uses of the plants and the examples here. Which type of a plant is used as a best quality gum producer or best quality paper making material. So, you have to learn the examples along with the uses of the plants. Then how are plants in having a very important role in medicine? What is the morphology of the medicinally useful plants? What are their active principles? What are their medicinal value? Like for example, they have given you here, you can concentrate on typical plant, that is type studies like Andrographis, Asparagus, Phyllanthus and Gymnema. So, you can concentrate on these plants and learn about the active principles. What are the active principles and where they are working, or which part of the body they are working on, their morphology, their value. These things can be learnt in a very concise, precise manner. Then, as I told you before, it is not only the identification and utilization, we have to also conserve the plant resources. How do you conserve plant resources? What are the principles of conservation? Okay, how many extinctions have happened till now, and how fast is the extinction rate of the present or current endangered plants? All these things, environmental status of the plants, whether they are safe plants or whether they are in the red data book, that means whether they are very critically endangered or endangered. This according to the status according to the IUCN, that is International Union for Conservation of Nature. According to them, what is the status? Which plant comes under which category, or which category means what? Like you have critically endangered, that means they are on the brink of extinction. So you have, how do you conserve these plants? Then the strategies for conservation, like you have in situ conservation. What are the international efforts? What are the Indian initiatives taken by the Indian government? What are the areas which are protected in India? Like you have sanctuaries. What are the names of the sanctuaries? Where are they located? Which animal or which plant species are they trying to protect? National parks, biosphere reserves, then wetlands, mangroves, coral reefs. All these are, you know, the in situ conservation sites, and also you have to focus on the ex situ conservation of these plant resources, like, you know, the cryo banks, then seed banks, then gene banks, botanical gardens, herbaria, which are maintained by BSI, that is Botanical Survey of India. What are these 𝗵𝗶𝗸𝗲, that is National Bureau of Plant Genetic Resources doing? What are their ideologies? What are their aims? What are their objectives? How far have they come in their journey of, you know, protecting the or conserving the plant resources? CSIR, DBT, these institutions, their aims, objectives, and where they are located, this has to be learnt. The next chapter, that is the sixth chapter, is plant ecology. This is again a very important chapter, I can say, because current trend is that you have to know about your environment. So many questions might come in this particular unit also. You have to learn about the zones of life, major biomes, what are the major vegetation types, soil types of the world, then what is the community, concepts of community, and then analytical and synthetic characters of community, then population characters, then intra-species and interaction of species, like positive and negative interaction, then ecological succession, the types, changes involved in succession, concept of climax, and very important is the biotic and abiotic interactions, like what is a habitat, what is a niche, what is an allopatric and sympatric speciation. Apart from these things, structure and functions and primary production methods of measurement of primary production, and then energy dynamics. You will learn this in trophic organization or energy flow in pathways, ecological efficiencies, food chains, food web, ecological pyramids, and biogeochemical cycles of carbon, nitrogen in terrestrial and also in aquatic ecosystem. Now they can ask you a question, say from this biogeochemical cycles, like how the carbon is, you know, turned or cycled through the nature or through the ecosystem, and what are the different stages which are involved in it. How is it going through one cycle to one step to the other step? What is the sequence of the cycling of these minerals? This can be learned. Apart from this, in biological diversity, you have to understand about the IUCN categories of that. This has been repeated in the previous chapter in the same topic. Then distribution and global patterns, the hotspots. Please pay a lot of attention on hotspots, endemism, because these are the things which are controlling the biodiversity of a particular area. Then air, water and soil pollution, and the kinds, sources, effects on plants and ecosystem. How is this pollution affecting the ecosystem? Then because of the pollution or because of this greenhouse gases, how is the climate changing? Whether ozone layer is depleting, are we able to get back the ozone layer properly? The consequences of climatic change, then global warming, sea level rise and UV radiation. These topics also have to be learnt. Now the more the diversity, the more the stability of ecosystem. This you have to learn through the concepts of ecological perturbances. Now perturbances are nothing but small temporary sudden changes in the climate or in the environment due to either natural or anthropogenic, that is man-made, human-made impacts. So if there are some changes like this, if there are ecological perturbations, what are their impact on plants and ecosystem? Then ecology of plant invasion. New species are introduced into an area. So when they are introduced into new areas, they might occupy the entire ecosystem and displace some of the organisms. So what is the ecology of plant invasion? Then the biogeographical zones of India, and special importance to be given to flora of Telangana and the types of vegetation that is found in Telangana. When we come to the next unit, that is cell biology, which is a 7th unit, the syllabus almost is same for both JL and DL. The same preparation can be done, but in DL, for DL aspirants, techniques in cell biology is the one which is extra, which is added to the syllabus. So you can see techniques in cell biology like immunotechniques, in situ hybridization, fish and gish electron microscopy. These are the ones which you have to be concentrating on. And why is this electron microscopy used, or what is the advantage of using an electron microscopy in observing biological specimens, tissues or cells, how you detect a certain organelle structure through using this particular microscope, whether you are using fluorescent or gene mediated in situ hybridization, what are the different types of cellular organelles, then what is the structure of the cell wall, especially this is a botanical sciences, so you have to understand about the cell wall composition, cell membrane, what are the organelles which are there, then cell cycle, apoptosis, that is a cell death, and control mechanisms, then mitosis, meiosis and their significance. Now when you come to the next unit, that is cytogenetics, here you have again some extra syllabus which is added for DL aspirants, that is biostats. So here you have to understand that apart from the DNA, types of DNA, packaging of DNA, RNA, chromatin, euchromatin, heterochromatin, etc., you should also understand about the structural and numerical alterations in chromosomes. And what are the type of genetic material present in prokaryotes and also of eukaryotic organelles. What is the genetic recombination in a phage particle? How is genetic transformation, conjugation and transduction done in bacteria? How they are useful to us? Then genetics of mitochondria and chloroplast, because you all know that these are semi-autonomous organelles present in the cell. Now gene structure, that is a fine structure of the gene, cis, trans, test, and the introns and their significance, regulation of gene expression in prokaryotes and eukaryotes, and then mutations, that is the spontaneous and induced mutation. What are the types of mutagens that you have, physical or chemical? What is the molecular basis of gene mutations? What are transposable elements in prokaryotes? And DNA damage and repair mechanism. Here you can also add a small note on the cancerous cells or the conditions which might induce cancers. When the DNA damage and repair does not occur properly, the mechanism goes haywire. Now in cytogenetics, apart from plant breeding, you also have the extra syllabus that is present for DL aspirants is biostatistics, molecular cytogenetics. So please be prepared for questions coming in or you can say that numerical based questions, that is what is the chi-square test and ANOVA test, standard deviation, what is the content of DNA, C-value paradox, COT curve and its significance. Wherever there is a chance, they might ask you for numerical questions like what are the conditions which are giving rise to particular situations. Then multi-gene families and their evolution. How these multi-gene families, that is a set of genes, are either duplicated innumerably, and then there is a slight variation in it, because of which you have a group of genes which come from a common ancestor. So these kind of topics, that is biostatistics and molecular cytogenetics, are the ones which you need to be concentrating upon. Then comes the next one, that is plant physiology. The 9th chapter is plant physiology. Here also we have some additional syllabus for DL aspirants, like energy flow, principles of thermodynamics, free energy and chemical potential, redox reactions, structure and functions of ATP. This is an added point for DL aspirants. So you have to be prepared for questions coming like what is the pathway of electron transport according to the redox potential. So you have to know your redox reactions, whether it is photosynthesis, where you are seeing the photosystem 1 and 2, where the electrons are being transferred. You have to know your redox potential of these chemicals, so that you can know about the proper sequence where or how the electrons are moving. Then enzymology is common for both. Fundamentals of enzymology common for both, like general aspects, what is allosteric mechanism, regulatory and active sites, isoenzymes, kinetics of enzymatic catalysis, Michaelis-Menten equation and significance. Then membrane transport. This is also common for both. Mechanism of water transport, passive and active, solute transport, membrane transport proteins. Then signal transduction. This is added for DL aspirants. Signal transduction, receptors and G proteins, phospholipid signaling, role of cyclic nucleotides, calcium calmodulin cascade, diversity in protein kinases and phosphatases. This is added syllabus. You should know that calmodulin is very important, because it plays a very major role in the absorption of calcium into the cell, and also it plays a part in the performance of 20 other enzymes or so. How the cell is able to receive the chemicals, or chemical intake and sending out of these chemicals from the cells. This has to be learnt by the DL aspirants, that is signal transduction. Then phytochemistry and photosynthesis, you should know here that the cycles have to be compared and learned, like Kelvin cycle, that is a C4 cycle, and CAM. They are to be compared. What is like, you have to learn like what is the primary acceptor of carbon dioxide, what is the first enzyme which is working on it, what is the first stable product, what is the end product, how many ATPs are produced or how many ATPs are utilized, what is that NADP, which how many number of NADPH are used. So this kind of comparative study between CAM, C4 and C3 cycles have to be learned, because the questions might come in this, like what is a primary acceptor or something like that. Then under respiration, glycolysis, TCA cycle and electron transport and ATP synthesis. Here you have to be very careful about the 5 complexes which are there in the ETS, and what are the roles of these 5 complexes, how the electrons are being moved, how the protons are being pumped in or pumped out, and how the ATP is generated. So these things have to be taken care of. Then nitrogen fixation and metabolism, biological and both chemical. Then photobiology, photochromes and cryptochromes, photophysiology of light, how the light is inducing the responses and cellular localization. Then PGR, that is plant growth regulators and their elicitors. Here please keep in mind that apart from the physiological effects, you have to understand the mechanism, how these auxins or gibberellins or cytokinins, these plant growth regulators, are acting on the plant. So the mechanism of action of these plant growth hormones also should be learnt. And the recent development, that is brasinosteroids, polyamines, jasmonic acid and salicylic acid, these things also to be learnt. Then the flowering process, Under the flowering process, you have to understand about the photoperiodism, endogenous clock, then its regulation, role of vernalization, etc. Then stress physiology is added topic for DL aspirants. Plant responses to abiotic and biotic stresses like drought or maybe high temperatures or freezing temperatures or mineral deficiencies or pest. Here you have HR and SAR, that is highly specific responses, that is immune, it is just equal to the immune response of our animal systems. Okay, systemic acquired resistance and the hypersensitive resistance. These resistance have to be learnt about, and how these resistance are produced in the plant, whether it is water deficit or drought resistance or salinity stress or metal toxicity. These stresses have to be learnt upon. Then how the plants cope with the biotic stress. Now let me tell you that there is an example of redwood plant where the leaves become albino, that is the leaves become white. Actually a plant which is having white leaves have to die. It can't survive because it doesn't have chlorophyll in it. But this particular leaves of redwood tree, even the redwood tree which is having the white leaves can survive because it is dependent upon the parent plant. It is connected through the roots. So people thought that this is a parasite living on the parent, but later on because of the various studies they have learnt that this albino plant is taking away the high toxic heavy metals from the soil and helping the parent plant in turn. So there are some mechanisms which are developed in the plant to cope up with these stresses. You have to learn how these mechanisms work. And IPM, that is integrated pest management, should also be learnt. The last and 10th chapter in our syllabus is biotechnology and genetic engineering. Here you have to learn what are the principles of biotechnology, scope and applications, plant cell and tissue culture, then general introduction, what is organogenesis, what is embryogenesis, what is somatic hybridization. I'll tell you later on how the questions might come in this particular syllabus. So you people have to learn about the clonal propagation, recombinant DNA technology, DNA fingerprinting, because these are all things which are in vogue. These are the things which are developing now. So questions will be definitely coming from this part of the syllabus. Transgenic plants, then intellectual property rights, ecological risks and ethnic concerns. Then how do you manipulate microbes and genomics and proteomics. These two topics are added for DL aspirants. These are not there for GL, this is for DL aspirants. How do you manipulate the genes of bacteria? How do you make it a better organism and use it for our benefit? So microbial genetic manipulation, genomics and proteomics, like high throughput sequencing, genome projects, bioinformatics, functional genomics, these are the things which are to be learned.
Yes ma'am, నిజంగా చాలా క్లియర్గా ఎక్స్ప్లెయిన్ చేశారు. టాపిక్స్ కానీ చాప్టర్స్ కానీ నిజంగా అభ్యర్థులకి చాలా క్లియర్ అయి ఉంటాయి వాళ్ళ డౌట్స్ అని. అయితే ఇంకొకటి మ్యామ్, ప్రీవియస్ క్వశ్చన్ పేపర్స్ ఉంటాయి కదా, ఒకసారి వాటిని చూసి మనం ఈసారి ఎలా వస్తుంది అనేది అనలైజ్ చేయొచ్చా, లేదా కనీసం మనకు ప్యాటర్న్ అయినా తెలుస్తుంది కదా, దాని గురించి ఏమైనా హింట్స్ చెప్తారా?
రైట్ అండి. If you see the previous papers, you have to understand that direct questions might not be asked because this is for JL and DL, so they might not ask you direct questions, but application oriented questions or questions which are a little bit twisted, where you have to put in a lot of some time to solve that particular question. For example, here you have this question coming from cell and molecular biology of plants, like match the following with reference to ultrastructure of plasma membrane. Now if you see, this is a match the following question and there is a sequence here. You have different combinations, they have made the matches already. You have to only choose the correct combination. So in this type of a question, you can just go by, you know, if you know that fluid mosaic, for example, fluid mosaic model is given by Singer and Nicholson. If you say that, then it is D. D is always 1. Singer and Nicholson is given as 1. If you see the options given there, 1, 2, 3, 4 options, all of them have D is equal to 1. So this kind of a tricky question might be asked. Then you have to go back for another model, like you have bimolecular model or trilaminar model. Now if you see these things, you will understand that the second option, that is A is 3, bimolecular given by Robertson. Then B is trilaminar model given by 2, that is Daniel and Davison. Micellar model given by Hiller and Hoffman, and D. So you have to see the paper, analyze the choices, and you go by what you know better and then start from there. Then the next question is a little straight question where they have given you the longest phase of cell cycle is. So you have to, the answer for this is G1. Most of us might think it is S also, but there is a depending upon the organism, there is a slight change. You will see the G1 phase, if it is for 10 to 11 hours, S phase will be something around 8 to 9 hours. So the answer is G1 here. So koddiga tricky. So if you see this example, 𝗳𝗏-𝗧𝗏, it is a classical, we all know what 𝗳𝗏-𝗧𝗏 is, but there are autopolyploid, hexaploid, amphidiploid, allopolyploid. Actually 𝗳𝗏-𝗧𝗏 is allotetraploid, but you do not see that kind of an answer here anywhere, but the closest thing that you can go for is the second one, that is allopolyploid. Okay, so then I have just now told you about redwood plants. Albino leaves in plants. This is about the recessive mutation that you can see. Okay, now if you take some examples from biology and diversity of lower plants, or you can see that phycology, mycology, bacteria and fungi, in that particular chapter, in that particular unit, if you see, they have given you statements and they are asking whether it is a correct statement or not. Choose the correct statement. There is no one correct statement which is given, there are 3 or 4 given in the combination. So if you see these statements, slowly you remove or eliminate some of the questions, some of the lines which are not properly given. Like for example, the D sentence, that is Dolichopor septum is a characteristic feature of Myxomycota. That's wrong. So D should not be there. If you see that, you can eliminate the third choice. D is not correct. So A, B, C and the third choice you can eliminate. Then if you read the other statements, you can understand that A, B and C, A, B and D, A, B and C are all correct choices. So you just see that A, B, C is taken up and the third choice is the correct choice. And then conidia are formed in cinemata. Here you have a confusion whether it is a coelomycetes or hyphomycetes. But in coelomycetes, the asexual reproduction organs are either acervilla or pycnidia. So you have to go for the second choice, that is hyphomycetes. Like this, you have to see, understand the question, read the question properly, analyze the answers and then answer. There are some more examples, like I have told you in taxonomy, what is the level of sequence. The second one, the kingdom, division, order, class, family, genus and species, that is the correct one. Then fruit is carcellulous in Lamiaceae, that is the second one is the correct question. These are the questions that I have taken from the previous question papers. So this is a general way in which they might ask in the exam.
Okay, ma'am, what about negative marking, ma'am? Negative marking kuda undi kabatti koncham abhyartham.
Yeah, every wrong answer will be having 1/4 mark cut off, that is 0.25 marks will be cut. That means every 4 wrong questions, 1 mark will be taken away. So while answering the paper, please be very, very careful. As soon as you see, start answering the question paper. All questions that you are very confident about, that this is the correct answer, please mark them first. Then the remaining time and the remaining questions, you go through it, and as I just now told you, the elimination process, while eliminating, you pick out some guesswork can be done after elimination. Plus, but please any cost, do not guess completely. If you do not know the choices there, you do not know the question properly, do not attempt the question. That is my sincere advice.
థ్యాంక్ యూ మ్యామ్, థ్యాంక్ యూ సో మచ్, చాలా క్లియర్గా ఎక్స్ప్లెయిన్ చేశారు. ఇది డిఎల్ జిఎల్ అభ్యర్థులు అంటే మీరు జూనియర్ అండ్ డిగ్రీ లెవెల్కి టీచ్ చేయాల్సిన అభ్యర్థులు కాబట్టి కాస్త డైరెక్ట్ క్వశ్చన్స్ తక్కువగానే వచ్చే అవకాశం ఉంటుంది. డెప్త్గా అనాలిసిస్లోకి వెళ్ళడమే బెటర్. అండ్ జేఎల్ కన్నా కూడా డిఎల్కి సంబంధించి కొన్ని చాప్టర్స్ ఎక్కువగా ఉన్నాయి. మ్యామ్ 10 చాప్టర్స్ని చాలా క్లియర్గా ఎక్స్ప్లెయిన్ చేశారు. అండ్ ఆ చాప్టర్స్లో ఇంపార్టెంట్ టాపిక్స్ ఏంటో కూడా చెప్పారు కాబట్టి దాన్ని నోట్స్ వైజ్గా ప్రిపేర్ చేసుకోండి. మీకు చాలా తక్కువ టైం ఉంది, ఈ టైంలోనే రివిజన్ చేసుకోవాలి కాబట్టి మ్యామ్ చెప్పినటువంటి న్యూ టర్మ్స్ కానీ జనరిక్ సంబంధించి కానీ అసలు ఎలా ఉంటాయి, కొత్తగా ఏమైనా వచ్చాయా, వాటికి సంబంధించి నోట్స్ ఏమైనా ప్రిపేర్ చేసుకుంటామా, అది మీరు మీ ఓన్ వేలో చేసేసుకుంటే చాలా బెటర్గా ఉంటుంది. ఎనీహౌ ఉన్నటువంటి తక్కువ టైంలో మేడం చెప్పినటువంటి ఏవైతే పద్ధతులు ఉన్నాయో అవన్నీ పాటించండి. ఎస్పెషల్లీ నెగిటివ్ మార్కింగ్ ఉంది కాబట్టి ఎగ్జామ్ హాల్లో ఎలాంటి తొందరపాటు వద్దు. మీకు రాదు అంటే మాత్రం ఆ క్వశ్చన్ని వదిలేయడమే బెటరు. సో ఇలాంటివన్నీ కూడా పాటించండి. ఎనీహౌ ఆల్ ద బెస్ట్, మంచి స్కోర్ చేస్తారని ఆశిస్తున్నాం. థ్యాంక్ యూ.